Very good day, students. So today we'll be talking about the functional evolution of the brain in part third of this lecture. So students, in the previous two lectures we had discussed about the two major parts. So we had divided this particular lecture into four parts. So the very first part was the medulla. Then we had discussed about the cerebellum, and now we are discussing in the third part. It is the midbrain. So now we are concerned with the midbrain in this particular lecture. So students, the midbrain is an integrating center in the case of the lower vertebrates. Now, as we have discussed in the case of the cerebellum also, that was the integrating center, and here the midbrain is also acting as an integrating center in most of the vertebrates. Now, what is the important feature in the midbrain? So actually it retains the nuclei of the optic system, and it adds to the auditory centers also in the case of the higher vertebrates. So midbrain is concerned with the two major systems. First is the optic system. So it is retaining all the nuclei of the optic system, and it is also functioning in you can say in the audition or the auditory part of many of the vertebrates. So midbrain has got some relation with the optic system. So we will be seeing in the later stages of this lecture that the midbrain is very much concerned with the vision of the animal, and to some extent it is also in relation with the auditory system of the animal. So students, if we talk generally about the animals, if I talk about the primitive animals just like cyclostomes, we talk about the early elasmobranchs or the bony fishes, or to some extent with the amphibians. Now if we take the structure of the midbrain, suppose this is the midbrain of the animal. So the dorsal part of the midbrain, I'm just taking the dorsal part as the red part of the brain or the midbrain. So this dorsal part of the midbrain is called as the tectum, and the lower part of the midbrain is called as the tegmentum. So sometimes in this particular lecture you will be finding this particular term tectum or tecta. Tecta is a plural word for tectum, and you may find tegmentum to some extent in this particular lecture. So these are the two parts of the midbrain. Tectum is the dorsal part and the tegmentum is the ventral part of the midbrain. Now out of the two, these the tectum and the tegmentum, so the upper part, which is the tectum, actually it is receiving the fibers from the optic tracts of the animal. So it is in direct relation with the optic lobes. So the tectum, it is highly in it is very much important part as far as the optic tracts are concerned or the vision of the animal is concerned. So in the next part of the experiments or the in this particular lecture we'll be talking about some of the experiments which will show you that the tectum is very important part in the vision of the animal. Now like the tectum, the second part, which is the tegmentum, actually it is also in relation with the optic muscles of the animal. So we know that the eye has got six types of muscles in the case of the, like suppose we talk about the fish. So we have different types of muscles which are like which are rotating the eye in a particular direction. So these optic muscles, they have got a connection with the tegmentum of the midbrain, or the ventral part of the midbrain. So what I want to say is that if you take whole of the midbrain as such, so it is in relation with the optic tracts or the optic muscles. So it is very much in relation with the vision of the animal. So here I am taking up the very first heading in the midbrain, that is the equilibrium and the vision. So students, we have seen that the midbrain is in connection with the vision. It is functioning for the vision of the animal. But to some extent, the midbrain is also involved in the equilibrium maintenance of the animal. So we'll be seeing that how it is like uh, in connection with the equilibrium also. So the very first experiment that I am taking is on the dogfish. So in dogfish, if the midbrain, the whole of the midbrain is removed, suppose it is removed, then what is the result of this removal of the midbrain? So will it be affecting the swimming? So here the swimming is not affected. So there is no effect on the swimming when we remove the midbrain from a dogfish. But the second thing which is seen by many of the scientists is that there is a poor sensory coordination. So the sensory coordination refers to some part it is in relation with the equilibrium of the animal. So if we have a poor sensory coordination, that means there is a loss in the equilibrium of the animal. So if we remove the whole of the midbrain, the tecta and the tegmentum, then there is no, actually there is no effect on the swimming as such. So there can be some of the posture control in the animal, but there is poor the sensory coordination. So senses are hampered to some extent. They are in relation with the equilibrium. Now after this, the scientists performed the second part of this experiment. So in the first part we removed the whole of the midbrain, but still in this part, in the second part, they removed only the tecta. So they removed the tecta of both the sides. So if I am just here, I am drawing the dorsal side of the midbrain. So we have actually depression in the midbrain. So if we see it from the dorsal side, we have got two tecta. So this is the tectum number 1 and this is the tectum number 2. So if we remove both these tecta from the dorsal side, what is the result of this experiment? So if they are removed, tecta are removed, so there is no vision as such. So this experiment, it is confirming the role of the tecta in the vision of the animals. So particularly in Scalidon or the dogfish, if we remove both the tecta from the dorsal side, there will be no vision in the fish. So actually uh, this is confirming the relation of the midbrain, especially the tecta, with the vision of the animal. Then again, scientists performed some more experiments to see the actual functioning of the midbrain. So what they did is that they stimulated the midbrain. So this big box is actually showing you the midbrain. So they stimulated the lateral sides of the midbrain. So they used very small micro electrodes which are particularly made up of gold. So they inserted these gold electrodes into the two lateral sides of the midbrain. So when the lateral midbrain was stimulated, so it resulted into the homolateral eye movements. So it is called as, so it showed the homolateral eye movements. So that simply means that the eyes were like they were moved towards the lateral sides, both the eyes. So the lateral midbrain stimulation, it resulted into the homolateral eye movements. So this experiment simply shows that if you like stimulate the midbrain, that will affect the eye muscles also, because we have seen that the tegmentum or the ventral side of the midbrain has got some relation with the optic muscles. So if you stimulate that particular part, we are not actually stimulating the tegmentum actually. So we are near to the tegmentum, we are in the central part of the lateral sides. So we are stimulating the lateral sides of the midbrain. So that is affecting the muscles or the optic muscles of the fish. Now students, we have an example of the goldfish. So particularly scientists have seen in the case of the goldfish that the tectum has some function in the learning process. So that means the tectum or the dorsal side of the midbrain has got some part, has got some function in the learning process. So if the fish learns some particular action, that we have to like go away from a particular thing, or we have to go towards a particular thing, so this kind of learning is concerned with the tectum part of the midbrain. So students, next example is of the ambystoma larva. So actually if we see the developmental stages in the ambystoma, so we like demarcate it as the stage 1, 2, and then we have the stage 3 of this larva, then we have the 4th stage, then we have the 5th stage, and so on. We have different stages of the development in this particular larva. So what the scientists they have seen is that uh, there is a part in the larva which is called as the bulbospinal apparatus, and this bulbospinal apparatus, it is in connection with the midbrain. So this is a part of the midbrain. So if the bulbospinal apparatus it is removed from the larva, so larva reaches a particular stage of the development which is called as the Harrison stage 40. So we have different stages like 1, 2, 3, 4, 5, and so on. So if you remove the bulbospinal apparatus, which is a part of the midbrain, the larva it reaches the stage 40, and this is called as the Harrison stage. So in this particular stage, what we have seen is that the larva becomes like comma shaped, just like this, and it starts moving like in a circular direction. So just like this, it is moving in a circular direction. So what I want to say over here is that if you remove the bulbospinal apparatus, it will be reaching the stage 40 and it will be moving in a circular direction. So that simply means that it loses its control on the locomotion. So this spiral movement or the circular movement, it is showing the loss of locomotory control. So that simply means that the bulbospinal apparatus has got some relation with the locomotion of the larval stage. So as we have seen that the midbrain, as we have saying that the midbrain has got a like a relation with the equilibrium. So here this experiment confirms that in ambystoma If you remove the part of the midbrain which is the bulbospinal apparatus, there is a loss of the locomotory control, or there is a loss of the equilibrium in the locomotion of the larva. The next experiment was done on the frogs. Now in frogs, when we like see the normal reproduction part of the frog life cycle, so they like go for spawning, they go for egg laying, and it has been seen that the tegmentum was the ventral part of the midbrain. So tegmentum was needed for the normal spawning movements. So I'm writing NSM for the normal spawning movements. So whenever the female frogs, they lay eggs in spawns, so they have to move in a particular, you can say, manner to make a spawn. So the normal spawning movements, they need the tegmentum part of the midbrain. So if we remove the tegmentum from the breeding frogs, the female frogs, the normal spawning movements, they are hindered, and that affects the spawning or the egg laying process in the frogs. The next experiment was done on the mammals. So especially I'm taking the case of the cats and the dogs. So students, what the scientists they have seen in the case of dogs and cats, actually, suppose we have the spinal cord, then we have the medulla oblongata, then we have the midbrain, suppose, and then we have the forebrain or the cerebrum, as such we can say over here. So in this part, if the whole of the cerebrum it is removed, leaving the midbrain, so what we have done is that we have removed whole of the part of the brain anterior to the midbrain. We have removed it. So like we have removed the cerebral part. So we have the midbrain intact. So this is the midbrain. So this experiment was done on the dogs and the cats. The brain is transected. It is cut anterior to the midbrain. So what they have seen is that like there is no rigidity of the cerebrum. So this is called as the decerebrate rigidity. So it is not seen in the cats and the dogs. So they are able to stand in this particular condition. So they can maneuver standing, and they can like stand on their legs to some extent. So that simply means that if the midbrain is present here, the midbrain is also present. So if it is present, they are able to like stand. So that simply means that if we have the midbrain, they can stand. So that is some part which is related to the equilibrium. So that is confirming the function of the midbrain in relation to the equilibrium in the mammals. So students, these were some of the experiments that were confirming the role of the midbrain in the case of the equilibrium and the vision in different animals. Now in the second part, I'll be taking the auditory reflexes in the case of different animals. So here we are discussing the auditory part of the midbrain, that how the midbrain is in relation with the auditory system. So auditory system, it refers to the system which is in relation with the sound. So first experiment that is like confirming the auditory reflexes in relation to the midbrain is on the cats. So what has been seen in the cats, that if the cats have given a particular sound and they perceive it, so what they have seen is, suppose this is the midbrain of the cat, and we have inserted two micro electrodes into the midbrain region. So what have been seen that if the cat is given a particular sound, these electrodes they show some potential. So whenever the sound reaches the ears, uh, I should not make this arrow towards the midbrain. Actually, suppose we have, these are the ears of the cat. So if we are putting the sound into these ears, so cat is subjected to some particular sound. So what has been seen is that these electrodes they are showing the potential. So if you have a potential, so we are having a particular graph which is showing the action potential of the sound. So that means if you give a particular sound to the cat, the midbrain shows an action potential graph. So that simply means that the sound system or the auditory system is functioning in relation with the midbrain. So this part demonstrates the visual responses which are in relation with the midbrain. So we have seen in the previous lecture that the midbrain is a very important part as far as the vision of the animals is concerned. So similar electrophysiochemical experiments were done on different animals. So as we have seen in the case of the auditory reflexes, that we get a like potential peak if we give sound to the cat. So in the same case, similarly, if we give light as a stimulus, if we give a light as a stimulus, they go to the eyes of the animal. And what has been seen that the midbrain is showing particular action potentials. So midbrain is giving you the action potentials or the graphs for the light. So this experiment also confirms the visual responses which are in relation with the midbrain. So students, if you combine the functions related with the midbrain to the equilibrium, to the vision, to the auditory reflexes and to the visual responses, so you can give a good idea of the midbrain or the evolution or the functional evolution of the midbrain in the case of different animals. So in the next lecture we'll be talking about the fourth part, which is the forebrain of the animals. Thank you so much.